Hello, everyone. On behalf of Invivoscribe, we welcome you to this corporate workshop organized by the Association for Molecular Pathology. My name is Julian D'Angelo. I'm the Senior Manager of the Bioinformatics Department at Invivoscribe, and I will be your moderator for today. Today's webinar title is Level Up Your MRD Testing, Leveraging Standardized and Sensitive AML Assessments. I'm very pleased to introduce our speakers for today. Our first speaker is Dr. Tony Sanchez. Dr. Sanchez received his BS in biochemistry from the University of Illinois Urbana-Champaign and went on to obtain his medical degree from Rush Medical College of Rush University Medical Center. After completion of a research fellowship at Medical College of Wisconsin, he completed a postdoctoral professional master's in bioscience management at the Keck Graduate Institute. He has since supported clinical field applications while working at the City of Hope, Thermo Fisher Scientific, and Canopy Biosciences. Currently, Dr. Sanchez is a technical account manager at Invivoscribe based in Chicago, Illinois, and manages the Mid-America territories. Our second speaker is Josh Wemmer, who is a senior bioinformatician at Invivoscribe located in San Diego, California. He received his master's in entomology with a focus on bioinformatics from the University of California, Riverside. As an undergraduate, Josh performed research at the University of Minnesota, and then after completing his master's degree, went on to work for the Salk Institute. As a part of the bioinformatics team at Invivoscribe, Josh has developed several variant calling, annotation, and QC pipelines to pair with our NGS-based assays, testing for hematological malignancies. He's currently a lead developer on our FUT3-ITD MRD and NPM1 MRD assay software. I'm now pleased to turn the time over to Dr. Sanchez for his presentation.
So we'll be covering today AML overview, speaking specifically about the etiology and pathogenesis. We'll talk about significance of FLT3-ITD and NPM1 variants. Then we'll delve into the topic of MRD monitoring, covering current established guidelines, significance of FLT3-ITD variants in terms of sensitivity, length, and frequency, as well as NPM1, sensitivity, subtypes, and frequency. And then we'll end with a conversation about assay design, key features, and advantages. So to begin with, I'd like to talk about AML. So basically AML is a cancer of the blood that has a distinct epidemiology representing the most common type of acute leukemia affecting adults over 65, characterized by the rapid growth of leukocytes in blood and bone marrow, with several known risk factors, including myelodysplastic syndrome, myelofibrosis, and aplastic anemia. Now, AML has been an important disease state to address because it represents an enormous unmet clinical need. According to the SEER database, those diagnosed with leukemia from 2013 to 2019, the average 5-year survival rate is a dismal 31.7%, representing less than half of all leukemias, of the leukemias taken across the board. Although through a better understanding of leukemogenesis, detecting of biologically important biomarkers with increasing sensitive diagnostics, and the development of therapeutics targeting those specific mutant genes, the aforementioned statistics could really be impacted. So for the purposes of our talk today, I'd like to limit the scope of the conversation strictly to genetic biomarkers that are important for pathogenesis of AML, beginning with an understanding of the classic paradigm of leukemogenesis. Really, classically, it's been seen as acquisition of a founding mutation by a cell population known as like an initiating event causing genomic instability, which then drives clonal evolution of AML founding clones, and then you're off to the races with frank disease. One very interesting and I think pivotal early study shed some light on the origin and evolution of AML, describing mutations relevant for AML pathogenesis. What's distinct, at least highlighted in this study and then supported later on by some clinical studies, was that many AML samples present with normal karyotypes and with genomic stability. So they tested a number of cell types, those that had known initiating mutations like PML-RARA, and those with unknown. They did whole genome sequencing and discovered a host of important initiating mutations like NPM1, DNMT3A, and so forth at high enough variant allele frequencies. There were a number that were detected, but these represented those of the highest frequency. And as I mentioned, they sequenced the whole genome of all of the samples from both cell types. And a couple things came out of the study, namely that both subtypes had overall a fair number of mutations that developed early on in the hematopoietic stem and progenitor cell population. And it wasn't really until you had this like 2 hits. So you had a hit of the initiating mutation followed by a progressive mutation like FLT3 later on. And it wasn't until then that you began to have founding clones, which then developed subclones, and then you had frank disease. What's interesting too is that the data suggests that most mutations found in the genome are random events that usually accumulate over time just due to age. And that really, sorry, one second. There we go. And that really, in some cases, it takes as little as 2 somatic mutations for you to develop AML. And that's demonstrated in this cell population, which is the M3 initiating mutation cell line that showed the presence of PML-RARA and FLT3. And it was only then that you began to develop AML. And so this, this suggests that as few as 2 somatic mutations can be important to develop the disease. That being said, advances in molecular techniques in the past, I would say 20 years, uncovered a complex, dynamic, and diverse genetic disease with multiple somatically acquired driver mutations, as you can see here, coexisting competing clones, and disease evolution over time, as highlighted in this paper, which was pulled from the 2017 European Leukemia Net recommendation paper. And so, in addition to highlighting all of the mutations discovered to date, mutations and translocations, et cetera, they did highlight 2 important targets, two drivers, particularly NPM1 and FLT3. And those I will address in a couple slides when we talk about the recommendations, the evolving recommendations that have been occurring since 2017. And so 2 important genes that I do want to emphasize are FLT3 and NPM1, which are probably known to most of the audience, but it's worth discussing very briefly that it's a monomer, and once it binds with the ligand, it dimerizes and then activates signaling pathways that are important for proliferation and survival, et cetera. Now, FLT3 represents 25% of newly diagnosed AML cases, and ITDs specifically, ITDs represent 25% of AML cases. ITD mutations specifically have an unfavorable prognosis, with the other class of mutations, TKDs or tyrosine kinase domain, occur less frequently and have unknown prognostic significance. So moving forward, we're really going to address just the ITDs in this particular talk. So here's just an overview of the gene. This is a wild type gene, and when you have insertion of ITDs within the juxtamembrane domain, the internal tandem duplicate transcript here becomes much larger and is detectable on fragment analysis. I'm going to de-emphasize TKDs, but those are also important in terms of being targets for pharmacologic agents, as are ITDs. The other gene to highlight that's also critical in our discussion is NPM1. NPM1 is an oligomer nuclear chaperone protein that's involved in many very important cellular functions, and a mutation that arises from the gene usually affects the exon 12, and it's involved in what you do eventually see is insertion of insertion here of anywhere from four base pairs or more at the region 960 at the 959, 960 nucleotide. You can have anywhere from type A mutations, which are known TCTC nucleotide insertions, and those represent 80%. Type B and D represent second and third most common, and followed by a few other rarer mutations. And what's interesting to note is that these frequently occur with various other mutations, including FLT3-ITD, and that's demonstrated here in this paper. It highlighted an important publication that showed that the presence of NPM1 alone was not detected in those with clonal hematopoiesis. However, when they co-occurred with other mutations like FLT3-ITD or DNMT3A, you had these mutations usually together promoted AML and suggested molecular synergism. And so in terms of diagnostics, when it comes to prognostics, it's advantageous to not detect this in isolation, but really in the context of other mutations, because these do seem to tend to occur together and play an important role in AML progression. So let's briefly cover the evolving guidelines that have been in circulation, some of which are probably already familiar to you. But I really want to limit the scope to diagnostic modalities and general recommendations surrounding the monitoring of disease after diagnosis, primarily using sensitive measures like MRD. So beginning in 2017, this ELN 2017, so ELN represents the European Leukemia Net, and it was a body that reviewed literature, a panel of experts that looked at all of the data and provided recommendations for AML for a host of things, not just diagnostics, but it involved diagnostic procedures, response criteria, and therapeutics. So really tried to encompass all of the care surrounding AML subjects. So in reviewing the material, I pulled out three relatively important topics that were addressed in this particular committee. The first being addressing the approach of MRD, specifically when it comes to modalities. And they isolated, or they emphasized flow cytometry, which has been mainstay for some time, but also the advent of molecular techniques like PCR or next generation sequencing based technologies. It was acknowledged that while these technologies can differ in terms of patient application and sensitivity, I think the committee realized that these would become more important over time as when you integrate baseline features with assessment of MRD to improve risk assessment. Second of all, response criteria was created, and I highlight that here, where a category was created for example, a complete response without MRD, or complete response with the presence of an MRD test that resulted in a negative result. And so this is beginning to show incorporation of MRD technology in terms of risk stratification. And finally, important work was done to identify risk stratification at diagnosis. This image here, this graphic here, represents the updated version, but it is not all that different from the one established in 2017, in which you had three categories: risk categories, favorable, intermediate, and adverse. And I want to point out that there are a fair number of genetic abnormalities that have been listed, but chief among them were NPM1 and FLT3. And while those have been removed from adverse in the most recent edition, they still exist in favorable and intermediate, still representing important biomarkers to monitor for newly diagnosed subjects. 2021 was the working body that examined MRD almost exclusively. It's a working party, and they really wanted to define some of the terms. For example, this is where the term minimal was changed to measurable residual disease. So also included some care in determining what positivity meant, what negativity meant. Also stress the clinical implications, specifically that MRD can be used as a prognostic or predictive marker, that it's an important monitoring tool to identify impending relapse, and finally as a surrogate endpoint for things like clinical trials. There's also some work to determine or to flesh out some of the technical specifications specifically for NGS when that was addressed in this publication. And so there was some work in determining, for example, what's the optimal nucleic acid, which was DNA in this case. What defines a good assay for NGS? For example, having sufficient read depth to discriminate signal from noise. Which specific genes to include, for example, not including germline mutations or mutations relevant to clonal hematopoiesis. And finally, including something like a standardized and harmonized bioinformatics pipeline. It's worth noting that they also addressed the limit of detection of the technology, which at this point was set at 10 to the minus 3. And as you'll see, it's going to evolve as the technology advances. The limit of detection does drop lower and lower. And then finally, using MRD as a prognostic and surrogate endpoint for drug tests, which is still very important. 2022 came along, and that's where they first and foremost improved this risk stratification, as I had mentioned. They also instituted some important recommendations for molecular MRD even further to include certain types of panels, emphasizing again which mutations to include and not include. And they also mentioned MRD testing as an important tool for decision making, specifically as a prognostic tool. And they incorporated numerous meta-analyses and multiple studies to justify these recommendations. And finally, response criteria. They basically took this and fleshed out a bit more so that it could be more clear. And then they dropped, in this case, a little bit of detection, specifically to referring to NGS, to 10 to the minus 4. Yeah, and finally, NCN and the NCCN guidelines sort of took these recommendations and incorporated them. The risk stratification in particular, identifying use of commercial panels if you're not an academic institution, and timing of assessment. So this is just a summary stating what I was mentioning above, that there have been a fair number of movements forward in incorporating, in fleshing out MRD as an assessment for prognostic implications. So the array of technologies, there are an array of technologies that do exist. I want to highlight a few of them, some of the tried and true ones that have been around for a number of years, and the ones mentioned specifically or emphasized rather, in the recommendations. I made mention of RT-PCR, flow cytometry, and NGS. And they do differ in terms of what cases they can apply it to and what sensitivity that they have. And as you can see, particularly for these last two, with the range of sensitivity much more improved relative to some of the earlier technologies. I believe these are the ones that have been moving forward in terms of clinical studies and application for prognosis. So the choice of technology really should address key aspects of MRD monitoring, like standardization assay with high sensitivity, as well as addressing specificity. So really, for MRD, you really do want the right test. And so that really begs the question, what are the best markers for for MRD monitoring. So a marker that's disease specific, stable over time, associated with relapse, and can have directed therapies against it. And also what makes for a good test for MRD, obviously sensitive, specific, and reproducible. But as for being internationally standardized, that's to say that it's important that a test can be used universally, so that's standardized in terms of workflow, particularly the bioinformatics portion, as well as the wet lab component, because you want to be able to use this test across institutions, particularly if this is serving as a surrogate endpoint for international clinical trials. So you want to have the same test across multiple trials across the world. And so to reemphasize, we already discussed that FLT3 ITD and NPM1 are important according to risk stratification. First and foremost, FLT3 present, we mentioned that it's 25%, present in 25% of new presentations of AML. We mentioned that it's a driver, significant driver, and late in leukemogenesis. You can see loss or gain of mutation at relapse, so it's important to monitor these regularly, and may only be present in subclones at initial testing. And finally, which I'll address in a bit, changes in ITDs between initial presentation and relapse can occur, and those ITDs do carry significance in terms of prognosis. Second, NPM1, also present in a high percentage of new AML presentations, ubiquitous among subclones and relatively stable, not typically associated with clonal hematopoiesis, and as I mentioned before, frequently co-occur with FLT3 ITD and other mutations. So next I want to highlight just a handful of publications that are important, that really emphasize these points, that these two gene markers are important, particularly for prognosis. So the first is a study that highlights the importance of FLT3 ITD as a prognostic marker in AML with impact on treatment outcomes. So of the subjects studied in this test, a number of them tested positive for ITD and underwent induction therapy, then tested once again when they were in relapse. And the presence of MRD was associated with increased risk of relapse, or 4-year cumulative incidence of relapse, not present here, but 75% positive versus 33% negative. What's shown here, though, is the overall survival sort of mirrors those results, in that those that were negative had a much improved overall survival relative to those that were positive. So it reveals that FLT3 ITD MRD is a strong independent prognostic factor. Second of all, another clinical study showed that where the secondary endpoint for this trial was looking at MRD survival of a randomized group of subjects pre and post transplantation. And as you can see here, the graph illustrates the progression-free survival of FLT3 ITD positive subjects randomized in this particular placebo group. As you can see, there is a stepwise decrease based on the limit of detection, or their variant allele frequency. So for every log of variant allele frequency that was increased, you see a subsequent decrease in survival, which is relatively significant. This suggests that limit of detection is an important factor when considering technologies to monitor progression after treatment. So not only does the presence of ITDs carry significant impact on survival, but so too does the length of those ITDs that are present. So in this study, adults with the mutation had their sample sequence and the ITD lengths and locations identified. The lengths ranged from 6 to 90 base pairs with a median of 39. And the genomic localization, probably 75% were in the JMZ region, 20% in the hinge, and then the remainder in the JMS region. And when followed when these patients were followed over time, post-treatment, then the treatment was transplantation, they found that for those whose ITD lengths were greater than 39 base pairs, they fared worse in terms of overall survival relative to the ITD length less than 39 base pair cohort. Yet another publication here that addresses the topic of microclones shows that ITD frequency, or the number of ITDs present in the sample, also has prognostic implications. So here I introduced the subject of microclones, which are smaller populations of cells that have different genetic makeup, basically have 2 or more FLT3 ITD clones present at diagnosis. And they can arise through various mechanisms, and they do matter because they provide valuable information about clonal evolution and heterogeneity of a tumor. They also harbor mutations that confer resistance to specific therapies. But at the very least, what this what this slide is showing is that the mere presence of microclones, or 2 or more ITDs, does confer a worse probability of survival relative to all the other subjects within this clinical trial. So, microclones may be associated with a decrease of survival. And up to this point, I've emphasized FLT3 ITD in the conversation of prognosis, but I do want to bring this point home that you want to test all of the AML associated variants. So in this study, they tested a number of variants, including NPM1, FLT3, ITD-H1, ITD-H2, and KIT. Most of which had FLT3 ITD mutations or NPM1 mutations. And among the AML subjects in first remission, the presence of either FLT3 ITD or NPM1 mutation in blood showed an association, an increased risk of relapse and worse overall survival compared with other AML associated mutations. And that's clearly seen here in this curve. And so I really want to mention that it is important to detect these 2 particularly important biomarkers when you're looking at new cases and when considering overall, when you're considering prognostic factors. And so with the understanding of the need for sensitive and with the specific and sensitive, harmonized and standardized tests for MRD, IVS has developed an assay that has been tested with research partners to really try to meet the need of the hematology oncology community to serve as a good tool for prognostication in AML. And that's the FLT3 ITD MRD assay and NPM1 MRD assay. The FLT3 ITD assay detects long ITDs up to 252 base pairs. The NPM1 detects 4 to 20 base pair inserts and reports them out as ABD or other. Both have an analytical sensitivity of 5 times 10 to the 5, with the possibility that some can be lower, but this is just strictly validated as 5 times 10 to the 5 minus 5. You can multiplex both assays onto the same flow cell, up to 24 indices, and you have the ability to multiplex with other lymphotract assays. Baseline samples not required. In addition, we use it's internationally standardized as an RUO kit, and also available as a clinical test through our CAP-CLIA lab PMM division for potential use as a surrogate endpoint. Finally, the assay is also available with the software, and both software packages are Dockerized and for ease of use in research, as well as locally installed and independent of the cloud. The workflow itself is pretty straightforward. You isolate material, DNA, from blood and bone marrow. You amplify through PCR and purify, normalize, placed on the sequencer. Analysis is completed by the aforementioned software, and then you have a final input that can be integrated into LIMS. And so with that, I want to hand this off to my bioinformatics colleague. So Joshua, please take it away.
Thank you, Dr. Sanchez. All right, I'll start sharing my screen here. Okay, so I'll be going over the software component of the presentation, starting with the FLT3 ITD MRD software. I'll first be going over the amplicon design as well as the variants that are reported. I'll then show a few example software outputs. And then I'll switch over to the NPM1 MRD software and present the same points. Again, the design and the variants reported, some example software outputs. And then I'll wrap it up with MRD solutions that the REO kits and software can provide. One of which is the different ways that it can be executed, including via the command line, and then also using the RESTful API that's built inside of the software. And so starting with FLT3 ITD MRD Dockerized software. This figure illustrates the region that is amplified in the FLT3 ITD MRD MiSeq assay. The juxtamembrane domain is represented by the light gray region. And within that, you can see that you have this darker gray region, and that's corresponding to the JMZ region of the juxtamembrane domain. And that is where you most commonly have ITDs inserted. The maximum theoretical detection size limit for ITDs is based on the insertion position within exon 14 of FLT3. So if you have an insertion position closer to the 5' end of exon 14, it can detect all the way up until 252 base pairs. If it's inserted closer to the end of the 3' region of exon 14, then it drops down 180 base pairs. Also, the software reports the minimum sizes for ITDs is 9 base pairs, and then for simple insertions, it's 3 base pairs. And then lastly, the software will only output results for in-frame insertions in ITDs. And so Dr. Sanchez gave a great presentation showing the clinical significance and prognostic value of having a very high sensitivity assay, being able to track all microclones in a sample and knowing the ITD lengths. And so this paper was already shown, but this from Liu et al. They were able to stratify patients based on the ITD lengths, that the mean of 39 base pairs, as Dr. Sanchez mentioned. And then additionally, a study by Dillon et al. In 2023 showed that the achieved detection sensitivity from the FLT3 ITD MRD REO kit and software can better identify patients with high risk of post-transplant relapse and death compared to less sensitive orthogonal methods. And so for all these points, there's strong justification for NGS-based testing for FLT3 ITDs to identify variants at lower limits of detection, tracking all microclones present, and knowing ITD lengths. And so I'll show a few example outputs of the software, just so you can get an idea of where this information is displayed and how it's formatted in the next few slides. So the software produces comprehensive results for each sample processed. And here I'm showing the final result TSV file for a single positive sample. And so you'll always have your input DNA amount in nanograms. This is provided by the user in the sample sheet CSV file. You'll also have your read depth. This is after pre-processing and filtering. And then you'll have your ITD result, which can be either positive, not detected, or undetermined. And then if you do have a positive result, you'll have your ITD size, the number of ITD supporting reads, and then your variant read frequency, which is simply just the number of ITD reads divided by the read depth. If you have a sample with multiple microclones, all of that information for each microclone is presented together like this. So you can see you have your different ITD sizes here, your number of reads, and then your VRFs. And then also you can see in this red box, you have this variant that's 174 base pairs long, and it's marked with this asterisk. And so that means it's a simple insertion rather than a tandem duplication. And this is defined in the glossary below as well. So I'll go over the differences between not detected and undetermined results. Here we have a not detected result, and this is when you don't have an ITD or insertion detected, and the confidence calculation is greater than 95%. So how do we get this confidence calculation? Well, in the software, we have this confidence calculator, and the question we're asking is, what is the confidence to detect a hypothetical ITD with an input VAF of 5e to the -5? And so what the calculator does is it takes in these two pieces of information, the input DNA amount and the read depth, which in this case are both high, and then provides us with this calculation of 99.99%. So very high confidence. It's above our 95% threshold, and therefore our ITD result is not detected. The software also provides this value here, which is the minimum ITD VAF detectable with greater than 95% confidence, and it's 5e to the -5. So this value is actually the lower limit for the software to report, and that's because this was the limit of detection we validated for use in our clinical lab, LabPMM, but this value could theoretically be lower from the software. So here's an undetermined result, which is when you have no ITD or insertion detected, and the confidence calculation is less than the 95% threshold. So again, you're taking in these two values here, the input DNA amount and the read depth. The input DNA amount is high, but the read depth is quite a bit lower. And so our confidence is also below our 95% threshold, 85.74% here. And so that's why this is an undetermined result. And then additionally, we have our minimum ITD VAF detectable with greater than 95% confidence is 6.8e to the -5. So one benefit of having this confidence calculator in the software is, for instance, if you have a low quantity of DNA in a sample that results in low read depth and therefore an undetermined result, it's possible to combine confidence results from multiple runs of the same sample to achieve the desired confidence. And the other thing too is it can help with troubleshooting with if you do have maybe a known ITD VAF, with this minimum ITD VAF detectable with greater than 95% confidence, this basically gives you kind of your new quote unquote LOD for the sample given this read depth. So depending on your use case, this could be useful information. So moving on to NPM1 MRD Dockerized software. I'll talk about the assay and software design and the variants reported, but first I just want to introduce this paper here from Ivy et al. In 2016. And what the researchers did is they looked at all NPM1 insertion variants in this region across 346 AML patients, and they counted the number of times they observed a specific variant, and then they ranked all of them based on their number of occurrences. So you can see at the top here, you have types A, B, and D, and they make up roughly about 90% of all insertion variants in this cohort. And you can also see that most variants in this specific region of exon 12 are 4 base pairs in length. Sometimes you have a little bit longer, but not quite as often. And so this is all to say that it's in this region of exon 12 that the NPM1 MRD MiSeq assay is targeting, and the software will report any insertion variant in this region between 4 to 20 base pairs in size. So I'll go through these next few slides a little faster than before because you can already see that the information displayed in the formatting of the results files are very similar to FLT3 ITD MRD software, but there are a few differences to note. So you have your input DNA amount and your read depth, of course, which will be used for the confidence calculation, just like for FLT3 ITD MRD software. And then you have your NPM1 MRD result, which can be positive, not detected, or undetermined. And then if you do have a positive result, you'll have information for the top NPM1 variant. And if your variant is type A, B, or D, you'll also be given annotations to go along with this. If it's none of those, it'll just say other. So here in this example, we have a type A insertion in column A. And then moving on to column B, you can see you have your RefSeq ID, your CDS, your amino acid change, your HG19 genomic coordinates, and then your number of variant supporting reads, your VRF. And then lastly, in column H, you have your contig-seq. And what the contig-seq is it's just the variant sequence detected in this specific region of exon 12. And then lastly, if you do have multiple variants detected by the software, it'll report the total number of variant supporting reads for all of those, as well as your cumulative VRF. In this case, there was only a single NPM1 insertion, so you can see that this information is actually the same as in the previous row. So for not detected and an undetermined result, the same information is displayed that I presented for FLT3 ITD MRD software. You can see here this not detected result. You have your 99.56% confidence, so we're above our 95% threshold. You can also see we have sufficient reads and DNA amount. For an undetermined result, you can see we're below our 95% threshold at 92.94%. You can also see that the read depth is quite a bit lower, and so therefore this result is undetermined. So because the software outputs for FLT3 ITD MRD and NPM1 MRD are harmonized, in this way you're and if you're using both the REO kits in-house, then this makes integrating the results into a database much easier. And so I'll move on to the MRD solutions that the REO kits and software can provide for you. And so one nice aspect of both FLT3 ITD MRD and NPM1 MRD is that they're Dockerized. And if you're not familiar with Docker, it's basically a platform that allows software to be built and deployed with ease through the use of containers. And all that's required is for you to have Docker installed, and then you can run any Dockerized software. And this means that the software is highly portable, since all of the dependencies the software needs is inside of the container. And so it can be run on any computational infrastructure, whether that is a server or in a cloud instance. Another nice feature is that there are two ways that you can execute the software. One is via the command line, which can be integrated into an automated workflow by using a workflow management system such as Nextflow or SnakeMake. And the other way is to execute the software via API requests to our built-in RESTful API called Drew. And Drew stands for Dockerized RESTful Executable Wrapper. And basically what Drew is doing is it's listening in on a specific port inside of the Docker container, and the user can then mount a port from the host machine. And this then allows the user to use an API client such as Postman to be able to queue up jobs seamlessly, check on run statuses, and then also update software configurations, just for example. And in the past, this is something we've helped clients with, automating off sequencer FASTQ files all the way to final results generation. And Drew was a key component for that. And so if that is something you're interested in, then we'd be happy to help you implement it. And so I'll just wrap up here with the main advantages that bringing in the REO kits and software in-house might provide you. So one, they're standalone. You don't need a baseline sample. Two, they're streamlined, which means faster turnaround times. Three, they're sensitive at a validated 5 × 10⁻⁵, at least for our clinical lab. This can theoretically be lower though, as far as the software goes. They're specific with reporting for all clones and VRFs, sequences, and lengths. And this is something I actually didn't mention earlier when I was showing the example outputs, but we do have in the supplementary info, or the raw results, if you will, the actual ITD sequences themselves, if that's something of interest. And then for both NPM1 MRD and FLT3 ITD MRD, the software does provide the insertion positions, if that's something of interest for you. They're just not in the final TSV results that I was showing, but they are available in the folders that are generated. And moving on, so they're accurate. The results are objective, especially with the reported confidence that you have if you don't have a detected variant. And they're also Dockerized, which makes them very portable. And so you can deploy them to any computational infrastructure, including the cloud. And then lastly, they're standardized for longitudinal studies and international collaboration. And so thank you for your time and attention. If you'd like to learn more about our comprehensive AML products and services, you can always email us at sales@invivoscribe.com, or you can scan the QR codes below if you want to learn about the kits and services that we discussed today. And while this presentation was focused on our REO kits and software, we do provide comprehensive AML products and services, including a multi-parameter flow AML MRD assay, all of which are standardized worldwide in our labs across the US, Europe, and China. These services include or utilize identical reagents and final interpretation by the same team of specialized hematopathologists in the US. The flow assay offers same-day turnaround times on specimens received in the morning. And then lastly, performance testing confirms it is the best flow-based AML MRD test available globally. And so with that, thank you again. I'd like to turn it over to Julian, and he'll lead us through the Q&A session.
Thank you, Josh and Dr. Sanchez, for your excellent presentations. We would now like to address some of the questions that have come in from today's listening audience. I will start with some assay-based questions, which Dr. Sanchez will help answer, and then move on to some of the software-based questions that Josh can answer. So Dr. Sanchez, the first question is, what sequencing platforms can the FLT3-ITD, MRD, and NPM1 MRD assays be run on?
Certainly, great question. So the platform that has been validated, the assay has been validated on the MiSeq platform, but theoretically should work on other Illumina instruments that use the roughly the same sequencing kit, the 2x300 base pair kits, sequencing kits.
Great, thank you. The next question is, can the FLT3-ITD, MRD, and NPM1 MRD assays be multiplexed with LymphoTrack assays?
Yeah, the simple, straightforward answer is yes. In fact, the assay was designed to be able to be multiplexed with other LymphoTrack assays. It just requires a bit of customization for the sample sheet available on the MiSeq. So the sample sheet CSV file allows for multiplexing of all three assays. Given that the FLT3 ITD and NPM1 MRD assays are dual indexed assays, whereas LymphoTrack are single index assays, there is a little bit of customization that needs to be done. It's not that difficult, and it's a simple matter of following up with the technical team to help work that out, and it's pretty straightforward.
When multiplexing, are there any other considerations for read allocation or anything?
Can you repeat that question?
When multiplexing, do you have to consider the amount of reads that are being allocated to each sample as you add more samples in the same flow cell?
Yeah, so there should be, I mean, just make sure that there are enough reads allocated per sample to avoid the undetermined calls.
Thank you. The next question for the assays are, what run controls are provided in the kits?
Right, so there are a number of controls that come standard with the assay. There's, for the FLT3 ITD MRD assay, there's a positive control. It's a 30 base pair ITD at a variant allele frequency of 5 × 10⁻⁵. And then for the NPM1 MRD assay, there's a type A insertion at 5 × 10⁻⁵. We also do provide negative controls that are pooled healthy tonsillar DNA. And it's also advised that whenever you do run an assay, that you do provide a positive control, negative control, and then an NTC.
Great, thank you. Next question is, what sample types are compatible with these two assays?
So the assay has been validated with blood and bone marrow. So those are the two sample types that are critical in order to be run on the assay, as well as extracted DNA. If this is brought in-house and the lab receives extracted material already via DNA, then that can be run as well.
Great, and on that, what is the minimum DNA input requirement for each sample?
700 nanograms.
Okay, great. Thank you, Tony. The next questions are related to the assay software. So, Josh, what is the minimum read depth required to avoid these undetermined calls at 5 × 10⁻⁵?
Yeah, that's a really good question. So after pre-processing and filtering, you need a achieved read depth of 157,600. But as far as like going into the analysis, we recommend that you have about 250,000 reads per sample allocated because you do expect a little bit of read dropout to get to that 157,600. And so this is to avoid an undetermined result if you don't have a variant detected.
Great, thank you. The next question is, how sophisticated do users need to be to run the Dockerized software?
I would say you don't need to have, a background in working on the command line or in bioinformatics or anything like that. We do provide a lot of extensive documentation to help new users. We have instructions in the form of READMEs as well as detailed videos. So I would say if you've never worked on the command line before, if you've never submitted API requests, you should be able to, with the helpful documentation, be able to get this software working. And then also, we're always here to help with the implementation and troubleshooting as well. So if you need technical support, we're happy to help out.
Great, thank you. The next question, Dr. Sanchez illustrated the wet lab and the overall workflow of the assay and software. And so this is more specifically for the software. How long does each software take to run?
Yeah, it does depend on your computational infrastructure, what kind of resources you have. For us, it takes about an hour, I would say, for FLT3 ITD MRD for full 24 samples. And then for NPM1 MRD, it's much faster. It's about, I'd say, 5 to 10 minutes to run. But again, this depends on your computational resources.
Great. And so on that, are there minimum computational requirements to be able to run the software?
Yes. So you need a minimum of 16 cores and then 64 gigabytes of RAM. And then for disk space, you need about 50 gigabytes per run. And that's including all of the FASTQ files that come off the sequencer as well, about 50 gigabytes of space.
Okay, great. Another question for FLT3 ITD MRD assay software is, can you sequence FLT3 large indels ranging and greater than 252 base pairs?
Yeah, so 252 base pairs is the maximum theoretical size for insertions ITDs. Yeah, that is the max.
I think with that, we've reached close to our closing time. So on behalf of AMP and Invivoscribe, we'd really like to thank everyone for your attendance and participation in the question sessions at today's webinar. We also once again thanks Dr. Sanchez and Josh Wemmer for their fine presentations. And again, please note this session has been recorded and these presentations will be made available on Invivoscribe's website. Thank you all.